name of Allah, who is the most beneficent and the most merciful biggest. I am Muqaddas Khalid from Batch 3. I am the student of UMT, and today I will discuss here Nobel Prize of 2020. Now we will start here from part 2 of our presentation, a crisper view of the 2020 Nobel Prize in Chemistry. The 2000 Nobel Prize in Chemistry was awarded to Emmanuelle Charpentier of the Max Planck Unit for the Science of Pathogens and Jennifer Doudna of the University of California for the discovery of the CRISPR-Cas9 genetic scissors that have revolutionized genome editing. While studying Streptococcus pyogenes, Charpentier discovered that a molecule called tracer RNA was part of bacteria's ancient immune system, CRISPR-Cas that cleaves viral DNA. Meanwhile, Doudna was deciphering the structure and biologic function of RNA enzymes and recognized the RNA-based surveillance system in bacteria. When they met at a scientific meeting in 2011, Charpentier and Doudna had a eureka moment, realizing that jointly they could harness the RNA-based complex to develop a novel gene editing system. Through a series of experiments published in 2012, they were the first to define an in vitro CRISPR-Cas9 system. That experiments published in 2012. They were the first to define an in vitro CRISPR-Cas system that not only could cut viral DNA but also could be programmed with an engineered guide RNA to cut any DNA at a sequence-specific site. Just as the discovery of restriction enzymes in the 1960s paved the way to an era of recombinant DNA technology. The CRISPR-Cas9 discoveries of Charpentier and Doudna have revolutionized our genome editing toolbox. In less than a decade, the applications of this powerful technology have exploded worldwide and have already led to advances in plant and animal engineering, personalized medicines, and promising clinical therapeutics. Here in this diagram, two sections also given. First one is A and second one is B. Clearly seen in this diagram that section A represents a guide RNA recognizes a genomic sequence and Cas9 cuts the DNA upstream of the protospatial adjacent motif sequence. Genome editing can lead to error-prone repair, small insertions or deletions, following non-homologous end joining that usually lead to lack of expression, or to define changings using a template for homology-directed repair. Additional strategies can be incorporated to precisely modify specific nucleotides or to target RNA. Here, you can see clearly section B. Section B explains several clinical applications for CRISPR-Cas exist or in development. In vivo application generally requires incorporation of the editing reagents in a vector, either non-viral such as a gel or in lipid nanoparticles, or recombinant virus such as adeno-associated virus, that is AAV. In vivo trials are ongoing to treat blindness, specifically helps simplex virus as well as HPV-driven cervical neoplasia and transthyretin amyloidosis, a rare congenital disorder that affects multiple organs due to amyloid deposits. Ex vivo applications include diagnostic applications such as for SARS-CoV-2 infection, as well as clinical therapies based on CRISPR-Cas modification of cells, which are then used to treat patients. Cell-based therapies to date include applications in hematological disorders and malignancies, renal cell carcinoma, and metastatic gastrointestinal cancers. From the CRISPR drive to the clinic. Now we will discuss here drive to the clinic. While the potential and wide-ranging applications of CRISPR-Cas continue to expand, CRISPR has already been at work in our dairy products, as the antiviral defense mechanisms maintain the consistency and acidity of yogurt cultures. In the agriculture industry, studies are underway to harness CRISPR to engineer a wide variety of plants to have better yields and superior resistance to pest and environmental stresses. Whether consumers will adapt to CRISPR modified crops remains to be seen, although there may be a lower threshold for acceptance of genetically engineered medications. Cannabis represents one such potentially lucrative target because engineering could allow more efficient extraction from single cell organisms engineered to secrete the cannabinoids into culture medium. CRISPR is also being evaluated for its potential to engineer livestock to eliminate the risk of deadly infections and to boost growth as well as to generate humanized organs that ultimately could safely be used for xenotransplantation. Meanwhile, diagnostic applications of CRISPR have emerged and continue to expand. CRISPR proteins can be designed as biosensors to identify a specific DNA or RNA target sequence. The assays are ultra sensitive, low cost, and fast. CRISPR has been used to identify various pathogens in clinical samples, including Zika virus, human papillomavirus, that is HPV, tuberculosis, and malaria. Perhaps one of the timeliest applications in an FDA-approved test for SARS-CoV-2 infection. Although the first CRISPR-Cas clinical trial was initiated only 4 years ago, there are currently 28 CRISPR-Cas studies listed on clinical trials government that are recruiting subjects. Although some of these studies are diagnostic or explore basic biologic questions, 21 aim to develop new therapeutics. The vast majority of the therapeutic studies use ex vivo strategies whereby CRISPR modified cells are infused to treat malignancies or lethal hematologic disorders. For example, studies of CTX001 produced by CRISPR Therapeutics aim to edit the BCL11A gene in order to increase the production of fetal hemoglobin as a potential treatment for beta thalassemia and sickle cell disease. CRISPR-based strategies are also being developed to treat positive solid tumors, hematologic malignancies, leukemia, and included too many such carcinogenic cancers, diseases, renal cell carcinoma, and metastatic gastrointestinal cancers. In less than a decade, researchers have used CRISPR-Cas9 to develop genome-edited crops, insects, genetic models, and experimental human therapies. Clinical trials are underway to use the technique to treat sickle cell anemia, hereditary blindness, and cancer. Doudna and Charpentier and others in the field have launched a generation of biotechnology companies aimed at developing the technique to achieve these goals. Now we will talk about safety and ethical considerations. If we talk about safety and ethical considerations, there are a number of safety concerns related to in vivo delivery of gene editing reagents. One of which relates to the potential of a harmful immune response because the Cas protein is derived from bacteria. It is likely highly immunogenic. In fact, in vitro transcribed guide RNA has been shown to trigger innate immune response, and anti-Cas9 antibodies have been identified in human serum. These pre-existing antibodies could neutralize the test reagent, thereby preventing any benefit, or worse, could lead to inflammation and tissue destruction. In addition, vectors used to encapsulate and deliver the editing reagents may incite an inflammatory response, which in terminally differentiated tissues, such as the retina, could lead to permanent tissue damage. However, the relatively immunoprivileged environment of retina, including tissue plants that limit drugs in distribution, a lack of direct blood supply, and presence of immune suppressive molecule was likely beneficial in the development of Luxturna, an AAV2 mediated gene augmentation therapy for congenital blindness that the FDA approved in 2017. Inherited retinal diseases thus represent a promising target for CRISPR based therapies, with several studies already in the pipeline. Besides immune response and the safety concern of in vivo delivery of CRISPR-Cas is the potential for off-target effects. Careful selection of guide sequences, incorporation of cell-specific promoters, and transient delivery of Cas9 may minimize these risks. The greatest concern is that a malignancy could result. By selecting tissue targets that can be directly visualized, the cervix, cornea, retina, in the first sets of in vivo CRISPR-Cas studies, regular non-invasive safety screening can be achieved. Finally, ethical concerns have already attracted the world's attention when CRISPR-edited human embryos were implanted and twins were ultimately born in China. Doudna was, in fact, the first person in the United States to hear about this work, when she received an email from the Chinese scientist planting the birth's announcement in 2018. The details of those gene editing procedures and the results are unknown, but the scientist conducting this work earned a 3-year jail sentence and a large fine too much dollars. This incident also stimulated much discussions about the need for international guidelines on human gene editing. Now we will discuss about the Nobel Prize in Chemistry in 2020 genome editing tools and their immeasurable applications for humankind. This year's Nobel Prize in Chemistry was awarded to two remarkable researchers, Emmanuelle Charpentier from France, now at the Max Planck Unit for the Science of Pathogens, Berlin, Germany. And the second scientist is Jennifer Doudna from University of California, who is also an investigator at Howard Hughes Medical Institute, San Francisco, CA, who have developed one of the most flexible and precise genetic scissors. The CRISPR-Cas9 system, at the time of the discovery, Emmanuelle Charpentier was studying the gene regulatory mechanisms of pathogenic bacteria to understand how they can acquire resistance to antibiotics and viruses phases, with the aim to find new therapeutic targets of infectious diseases, while Jennifer Doudna was leading the RNA structural biology field from the early 2000s. Their passion and curiosity for life science led them to strike up a highly fruitful collaboration building on their complementary background and findings, which ultimately opened the path to the discovery of CRISPR-Cas9 system as genetic scissors, since the Human Genome Project began in 1990. Scientists, especially in the medical field, have been trying to identify the function of genes underlying human diseases, gene targeting in mice, and RNA interface, both of which were awarded the Nobel Prize in Physiology and Medicine in 2006 and 2007, have been contributing greatly to breakthroughs in the understanding of mechanisms of diseases. In the meantime, scientists have also tried to develop technologies that can edit the genome of cells and organisms to understand the function of genes of interest, with the ultimate dream of correcting genetic errors. Engineered nucleases such as finger zinc nuclease ZNFC and transcription activator like effector nuclease were the first versatile tools to be developed for genome editing. But the great technological leap forward occurred with the unraveling of the CRISPR-Cas9 system in 2011 to 2013. What is the purpose of precise genome editing technology? What is the main purpose? Two scientists who pioneered the revolutionary gene editing technologies are the winners of this year's Nobel Prize in Chemistry. The Nobel Committee's selection of Emmanuelle Charpentier, now at the Max Planck Unit for the Science of Pathogens in Berlin, and Jennifer Doudna at the University of California, Berkeley, puts an end to years of speculation about who would be recognized for their work developing the CRISPR-Cas9 gene editing tools. The technology allows precise edits to the genome and has swept through laboratories worldwide since its inception in 2010. It has countless applications. Researchers hope to use it to alter human genes to eliminate diseases, create hardier plants, wipe out pathogens, and more. The ability to cut DNA where you want has revolutionized the life sciences, said Pernilla Wittungsted, a biophysical chemist and a member of the Nobel Chemistry Committee, at the prize announcement. The genetic seizures were discovered just 8 years ago but have already benefited humankind greatly. Unique aspect of CRISPR-Cas9 genome editing has been the ease and versatility of technique, he adds. CRISPR-Cas made this so much more readily acceptable, says Collins. There is no molecular biology laboratory that I know of that has not started to work with CRISPR-Cas. Now we will discuss application of CRISPR-Cas system in cancer research. How they play an important role in cancer research. Cancer initiation and progression involved in mutation and dysregulating expression of a series of genes including oncogenes, tumor suppressive genes, chemo-resistant genes, metabolism-related genes, and cancer stem cells-related genes. The ultimate goal of cancer treatment is to restrain tumor growth and progression by specifically correcting mutation and restoring expression of dysregulating genes. The CRISPR-Cas9 gene editing system has been widely used in the basic research of cancer, and some encouraging progress has been achieved. Cancer is a potentially fatal disease that accumulates multiple genes and alters epigenetics throughout the genome. The mutation of genes in cancer usually drives the proceeding of cancer and impacts the future of tumorigenesis. During the past 2 decades, a large number of genes related to cancer initiation and progression have been identified by high throughput sequencing technology. Based on these progressions, gene editing technology holds a big promise for cancer treatment via adjustment of the expression and correction of mutations of genes, which may lead to further breakthroughs in the field of precision oncology. What is oncogenes? Oncogenes are regulated differently from normal genes and can promote malignant transformation of normal cells. The CRISPR-Cas9 system provides a valid measure for deletion interfering with the expression and changing the activity of oncogenes, thereby inhibiting tumor growth. The CRISPR-Cas9 mediated knockout of CD133 down-regulated vimentin expression in colon cancer cells, significantly reduced cell proliferation and colony formation, and showed significant inhibitory effects on cell migration and invasion. Knockdown of MYR3064 using CRISPR-Cas9 technology significantly inhibited the proliferation, invasion, and tumorigenic capacity of pancreatic cancer. Besides, using CRISPR-Cas knockdown of oncogenes mutant EGFR alleles, the growth and proliferation of lung cancer cells lines H1975, A549, and H1650 were found to be inhibited, and the tumor size of xenografted mice implanted with H1975 or A549 cells was reduced. Knockdown of the FAK gene in NSCLC cells with KRAS mutations using CRISPR-Cas9 resulted in detectable DNA damage and increased sensitivity to radiotherapy. Furthermore, the CRISPR-Cas9 mediated deletion of the E3 ubiquitin ligase UBR5 in an experimental murine model of triple-negative breast cancer (TNBC) remarkably inhibited tumor growth and metastasis in vivo. These studies indicated that CRISPR-Cas9 gene editing system is an effective tool for identifying oncogenes and evaluating the therapeutic potential of oncogene targeting. Here in this diagram, 4 sections A, B and C. This is the CRISPR-Cas9 in cancer research. Section A is about DNA-based knockdown, knock-in or knock-out. Section B represents library. Section C represents RNA targeting. And Section C here represents the introduction and visualization. In next step, we will discuss all 4 sections A, B and C in detail. CRISPR-Cas9 mediated knockout or knock-in was applied to identify and verify a functional gene including oncogene, tumor suppressor gene, chemoresistance gene, metabolism related gene and cancer stem cell related genes. And section B, CRISPR-Cas9 library screen for drug target and functional gene. Section C represents application of the CRISPR-Cas13 system for RNA targeting. And section D represents in vivo biotinylated dCas9 protein was used for interaction research. And dCas9 proteins fused with fluorescent markers such as GFP was used for imaging analysis. Is it clear? Now we will discuss about cancer diagnostics. Application of CRISPR-Cas in cancer diagnostic. Early detection and treatment of cancer can reduce mortality and improve the life quality of patients after treatment. Although several techniques are widely used for cancer detection, they need improvements in terms of sensitivity, specificity, and speed. Therefore, identifying sensitive genes through genetic diagnosis is key to the prevention of cancer. To overcome these problems, Gutenberg et al. Established a CRISPR-based diagnostic system called Sherlock Specific High Sensitivity Enzymatic Reporter Unlocking. This system consists of the RNA-guided RNase Cas13a and a reporter signal which showed high sensitivity in mutation detection of BRAF. In this diagram, we will clearly see application of CRISPR-Cas9 gene editing tools for diagnosis and for the therapies of cancer. Here also 4 sections in this diagram. A, B, C, D, E, 5 sections. A section diagnostic, B section CAR T cell, C section PD1, PDL1 and D section represents combating carcinogenic virus and E section represents animal modeling. Clear? And one by one we will discuss here the detail of all these sections. A, using CRISPR-Cas9 based diagnostic system Sherlock and detector for detecting cancer. B section represents CRISPR-Cas9 edits immune cells in vitro and then these cells were administered to patients to combat against cancer. Here you can see, okay. C section, knockout of inhibitory receptors PD1 by CRISPR-Cas9 technology improves the efficacy of cancer immunotherapy. Section D represents viral genome specific Cas9 sgRNA eliminates oncogenic virus. establishing in vivo tumor model with multiple gene mutations with CRISPR-Cas9 gene editing tools. Applications of CRISPR-Cas9 in cancer therapy. Before starting this topic, we will again repeat this diagram and its explanation that is given its above. Here HPV virus also explained, and we discuss our diagram with detail in this section. Okay, application of CRISPR-Cas9 in cancer therapy. In addition to making rapid progress in basic research of oncology, CRISPR-Cas mediated genome editing also has broad prospects in cancer treatment. Tumorigenesis is a multi-step process involving complex interactions between cancer cells and the host immune system. The combination of the CRISPR-Cas technique with cancer immunotherapy and its application to combating carcinogenic virus infection offers great promise. If we conclude our whole assignment, then we will discuss about all these genetic advanced technology and what we will learn about these all terms and explanations. So we will conclude our topic. The discoveries by Charpentier and Doudna demonstrate the revolutionary impact that can result from basic research. What once may have seemed an esoteric enzyme of the microbiology world is now a household name that has led to simple, efficient, and cost-effective genome editing and groundbreaking advances in biology and medicine. This Nobel Prize also reminds us of the importance of interdisciplinary scientific collaboration of eureka moments made possible by human interaction, and of the power of scientific curiosity and ingenuity. While CRISPR based genome editing has the potential to treat inherited disease, the power of rewriting the genome has also raised concerns about what is morally acceptable. The future of this exciting field, therefore, demands a careful balance of scientific feasibility and ethical vigilance. In just a few years, the CRISPR-Cas has emerged and advanced rapidly as a stable, efficient, simple, and extensively used gene editing technology. Actually, the CRISPR-Cas9 has fundamentally impacted many fields, such as agriculture, biotechnology, and biomedicine. But no field has felt a more profound impact than cancer research, as evidenced by the accumulating data in the fast-growing publications. The capacity to implement the genome rapidly and accurately opened the windows for a new and more elaborate outlook on the mechanisms of tumorigenesis and progression. More importantly, CRISPR-Cas9 gene editing technology hold big promise for cancer therapy. However, several challenges remain before this technology can be used in clinical treatment of cancer safely and efficiently. Thank you so much. Allah Hafiz.